In this session, we are going to study about the anatomy of dicot and monocot root. So let us start. First of all, let us learn about the anatomy of a dicot root. So if we take the transverse section of a root and observe it under the microscope, then we will find all these different layers and tissues which we are going to understand now with the help of a diagram. Alright, so let me draw. So if we start with the outer layer, then we know that the outer layer comprises of the epidermis, right? Whether it is a root or it is a stem. So this will be the outer layer, which is usually the epidermis. But in case of root, the epidermis is called epiblemma. Alright, and we also studied that the epidermal cells of the root, they expand and form unicellular hair like this. So some of the epidermal cells will form root hairs. Alright, so this is epidermis or epiblemma. The epidermis of roots are called epiblemma. Then there will be root hair present on epiblemma. They help in the absorption of water and minerals. Right, we studied about it when we were learning about the epidermal tissue system. Then below the epidermis, we will find cortex. The cortex consists of parenchymatous cells which are thin-walled and they have intercellular spaces between them. So we will find several layers of parenchymatous cells which are thin-walled and having intercellular spaces between them. This is called cortex. Cortex is present below the epiblemma or epidermis. Alright, so this is cortex. Now below the cortex we will find the innermost layer of the cortex is called endodermis. So this forms the endodermis, which is the innermost layer of the cortex, and it is single layered and made up of barrel shaped cells. Endodermis is single layered and is made of barrel shaped cells which are closely packed like this. Now this endodermis has a deposition of water impermeable waxy material which does not allow the water to pass through it. So this special type of deposition inside the endodermis is called Casparian strip. there is a deposition of suberin. Suberin is a water impermeable waxy material which is deposited inside the endodermis and forms Casparian strip. Alright, so this is endodermis, which is the innermost layer of the cortex, and inside the endodermis there is Casparian strip, which has a deposition of suberin. Suberin is water impermeable and thick material which does not allow the passage of water through it. Alright, then inside the endodermis we will again find a few layer of parenchymatous cells which will be thick. The walls will be thick and there will be no intercellular spaces between them. Alright, so a few layer of thick walled parenchyma cells with no intercellular spaces between them. This layer is present below endodermis and it is called pericycle. Now we have studied that the root sometimes have lateral roots, right? So these lateral roots arise from the pericycle. So the parenchymatous cells can enlarge and give rise to a lateral root like this. Alright, and also in the case of dicot, since we are studying about the dicot root, when the secondary growth takes place, when the cambium is formed, so that formation of vascular cambium occurs inside the pericycle only. Alright, so this was about the pericycle. Then inside the pericycle we will find the vascular bundles, and we studied about how these vascular bundles are arranged inside the root. In the previous lecture we studied about it, right? So the vascular bundles are arranged radially. That means xylem and phloem will be found alternately on different radii. So in case of dicot roots, usually 2 to 4 xylem and phloem bundles are found. So this will be the xylem. And we also studied that in roots the metaxylem is towards the center and protoxylem is towards the outside or towards the periphery. Similarly, let us draw the phloem on different radii. So these are the patches of phloem. Alright, so we can see that the xylem and phloem are arranged radially in an alternate manner on different radii, and the xylem and phloem, the number of xylem and phloem bundles are usually 2 to 4 in dicot roots. Alright, now between the xylem and phloem the parenchyma cells will be present, and these parenchyma cells form conjunctive tissue. So what is a conjunctive tissue? The tissue present between the xylem and phloem, which is made up of parenchyma cells. So this is conjunctive tissue. Now at the center of the root or the stem, the central portion, the innermost central portion, this portion is called pith. So in dicot roots, the pith is very small or inconspicuous. pith very small or inconspicuous. Inconspicuous means not easily noticeable. So the pith is not well developed in case of dicot roots. Alright, and the area from the pericycle to the pith, that is pericycle plus vascular bundles plus pith, together forms stele. So let me write here. What is stele? Stele is pericycle plus vascular bundles plus pith. So these three layers, pericycle, vascular bundle and pith, are together called stele. Alright. So from this portion, this is the pericycle, then this portion is the vascular bundle and this portion is the pith. So from here to here it is stele. Alright. So this was about the anatomy of dicot root. I hope it is more clear to you with the help of the diagram, all the layers from the epidermis to the pith. Alright, so let us move on to the anatomy of monocot root. So if we talk about the monocot root, then the monocot root will be having similar layers to that of the dicot root. That is, everything will be the same except the vascular bundle and the pith. Alright, so let us learn about what is different in monocot root if we compare it with the dicot root. So there are just three differences, otherwise the diagram is the same. So if we talk about monocot root, in monocot root, the xylem bundles are usually more than 6, which is called polyarch. Xylem bundles are more than 6. In dicot, we studied that the xylem and phloem bundles are usually 2 to 4 in number. But in monocot roots, the xylem bundles are usually more than 6, and this is called polyarch. So this is the first difference. Alright, so in case of dicots, 2 to 4, in case of monocots, usually more than 6 xylem bundles are present. This is the first difference. Then the second difference in monocot root is that the pith is large and well developed. In case of dicot root, we studied that the pith was inconspicuous. That means pith was very small and not well developed. It was not easily noticeable, right? This portion. But in case of monocot root, it will be large and well developed. This is the second difference. Now the third difference, there will be no secondary growth in monocot roots. So there will be no formation of cambium. So any tissues which were giving rise to cambium in the dicot root, that is the pericycle. Right, we studied that the pericycle, the cells of the pericycle forms lateral roots as well as vascular cambium, which causes secondary growth in dicot roots. But since in monocot root there is no secondary growth, so there will be no formation of cambium in any of the tissues. Alright, So these are the three major differences. If we compare monocot and dicot root, the structure would be similar. The anatomy of the monocot and dicot root would be similar in all respect except these 3 points. In dicot roots, the xylem and phloem bundles are usually 2 to 4 in number, and the pith is very small or inconspicuous, whereas in case of monocot roots, the xylem bundles are usually more than 6, which is called polyarch, and the pith is large and well developed. Besides that, in monocot root there will be no secondary growth, but secondary growth will be seen in dicot roots. So I hope the anatomy of monocot root as well as dicot root will be clear to you. So all these layers from the epidermis to the pith, that is epidermis, cortex, endodermis, pericycle, vascular bundles and pith, these all will be similar in monocot and dicot root except the number of vascular bundles and how well the pith is developed. Alright, so this completes the anatomy of dicot and monocot root. In the next lecture, we will study about the anatomy of dicot and monocot stem. Alright, thank you so much.